Restriction enzyme, also called restriction endonuclease, is a protein produced by bacteria that clips DNA at specific sites along the molecule. In the bacterial cell, restriction enzymes clip foreign DNA, thus eliminating infecting organisms. Restriction enzymes can be isolated from bacterial cells and used in the laboratory to manipulate fragments of DNA, such as those containing genes. For this reason, there are indispensable tools of recombinant DNA technology or genetic engineering. Restriction enzymes are traditionally classified into four types on the basis of subunits composition, cleavage position, sequence specificity, and cofactor requirements. Type 1 enzymes are complex, multi-subunit combination restriction and modification enzymes that cut DNA at random far from their reconnection sequences. Type 2 enzymes cut DNA at defined positions close to or within their recognition sequences. They produce discrete restriction fragments and distinct gel binding patterns, and they are the only class used in the laboratory for routine DNA analysis and gene cloning. Type 3 enzymes are also large combination, restriction, and modification enzymes. They cleave outside of the recognition sequences and require two such sequences in opposite orientations within the same DNA molecule to accomplish cleavage. They rarely give complete digests. Type 4 enzymes recognize modified, typically methylated DNA and are exemplified by the MCRPC and MRR systems of E. coli. There are several things that should be considered when using restriction enzymes. Some of them are pH, because most restriction enzymes are used between pH 7.2 and 8.5 as measured at the temperature of incubation. pH values outside of the optimal range may lead to star activity. Magnesium concentration, because restriction enzymes requires that concentration as the only cofactor. Salt concentration. Restriction enzymes are diverse in their response to ionic strength. Ones might be inhibited by salt concentrations higher than 20 micromolar. BSA, which is used in restriction enzyme storage buffers and is added to some digestion reactions to stabilize the enzyme. Glycerol, which is added to restriction enzyme storage buffers to prevent freezing at minus 20 Celsius degrees. Incubation temperature. Most restriction enzymes show maximum activity at 37 degrees. Generally, the incubation temperature for the enzyme reflects the growth temperature of the bacterial strain from which it is derived. Volume, because viscous DNA solutions inhibit enzyme diffusion and can reduce enzyme activity. For the generation of a restriction map of a vector, it is necessary to have the sequence of interest. There are various programs to generate this restriction map, like SnapGen or NapGather. We'll do this in NapGather because it's free. The sequence of interest will be inserted in the space, and depending if it is linear or circular, we will pick that choice and click Submit. Next, we will be generating a map of the vector, and we can see all the restriction enzymes that it contains. For the digest of this vector, we will pick on the option of Custom Digest. Next, we will choose the restriction enzymes that we want, and next we will click Digest. And we can see a restriction map with the restriction enzymes of interest. Here we can also make a simulation of an agar gel. So we click on View Gel, and next we get an electrophoresis simulation with all the possible cuts, and we can select certain characteristics like gel type and the percentage of agarose, the marker, and the DNA type.
In order to make the practice, a mixture of different substances need to be prepared in order to make the characterization or the restriction of the plasmid. And the substances need to be added in the following order. First of all, the water. And then the buffer need to be added. In this case, net buffer 3.1 at 10x concentration was used. BCA also need to be added, but with the buffer 3.110x, it is not necessary to add it separately because it included. Then the plasmid has to be added to the mixture, and finally the restriction enzymes. As the practice requires 3 different reactions, that means that the first reaction need to contain the restriction enzyme SCA1. The second reaction would contain the restriction enzyme SHO1. And the final reaction, that means the third reaction, need to contain both of the previous enzymes. That means that it has to contain the SCA1 and SHO1 in order to make the restriction. It's necessary to ensure that all the volumes added to the reactions are mixed together in order to obtain good results and to obtain a good electrophoresis gel. As said before, the restriction enzymes were added at the final step. Then the mixtures or the reactions were incubated for 1 hour in order to ensure the action of the restriction enzymes in the plasmid. Finally, an electrophoresis was prepared and run in order to ensure the cuts or the restriction of the plasmid. Once the electrophoresis was run, the following patterns were obtained. In the first lane, we can see that the marker was loaded correctly. In the second lane, there are different defined bands that may represent the isoforms of the plasmid. In the third lane, there are more bands than in the original plasmid. There is the second lane. In the fourth lane, there is only one remarked band. And finally, in the fifth lane, there are some other marked bands and one band that is at the bottom of the gel. In the gel, there are blots in the walls that mean protein contamination. Also, there are degraded bands all along the gel that means that the genetic material was degraded. Also, there are marked blots in the electrophoresis gel that can represent RNA contamination or some other genetic information contamination. Also, there are marked blots in the electrophoresis gel that can represent RNA contamination or some other ribonucleic acid contamination. Also, in the bands we can see a degradation pattern or patterns that make oscillations in the bands. It can represent the presence of salts in the samples. In the second lane, we can see different bands, at least 3 or 4, and those can represent the isoforms of the plasmid. In the third lane, we can see more bands than in the original plasmid. These can represent the cuts of the first restriction enzyme, that is SCA1. But there is an error because this enzyme cuts twice in the plasmid, and here we can see more than 2 bands. In the fourth lane, we can see only a marked band, and this represents the cut of the restriction enzyme SHO1. This restriction is correct because SHO1 only cuts one time in the plasmid. And finally, in the fifth lane, we can see 3 bands that represent the cuts of both enzymes. That means that SCA1 and SHO1 cut at the same time. We can see 3 bands, but the second one is bigger than the other ones, and this can represent different bands overlapped. In this lane, we can see that the action of the restriction enzymes is correct because there are different cuts, and also there is one final cut that is smaller than the other one.
In this practice, we could digest a plasmid and visualize it in an agarose shell. It is important to verify the integrity of the enzymes used because it can cause trouble in the visualizing of the gel, as well as do unexpected cuts. The digestion is of importance because it can show us the correct direction of the insert, as well as obtaining the fragment of interest for another purposes.